Hello, everyone. Welcome to the de novo analysis of single-cell data breakout session. My name is Kevin Rose, and I'm a graduate student in Dr. Rona Hartzano's lab at the University of Maryland School of Medicine. In this session, we're going to be walking through the single-cell RNA-Seq workbench features in the gEAR. To navigate to the single-cell RNA-Seq workbench from the gEAR's homepage, simply find the box entitled Single-Cell RNA-Seq Workbench and go ahead and click Launch Workbench. This brings us to our first step. The first step is choosing the dataset you want to analyze. We're going to be analyzing a publicly available dataset generated from the cochlea of P2 mice. So to simply find this dataset, we click on the drop-down menu on the left-hand side. We go to Public Datasets. We scroll down until we find a dataset called HRP, Cochlea Single Cell P2, Hartzano/Ament. Now, to reiterate what the title says, this is a P2 cochlear dataset, of course, single cell. Now, this brings us to the first step of the actual de novo analysis. You can see that we want to be on New in the drop-down analysis window of stored analyses because we are indeed doing a de novo analysis. You can also see at the bottom left here are the different steps that we will be working through sequentially. The first step in any single-cell RNA-seq dataset is to filter out outliers. Outliers could be cells with hardly any genes sequenced or genes that were identified in only a very small number of cells. These data do not meaningfully contribute to our analyses, so we will filter them out. The data settings are set to exclude cells with fewer than 300 genes and exclude genes found in fewer than 3 cells. So to apply these default settings, we can simply click on the tick boxes next to exclude cells with less than 300 genes and exclude genes that are in less than 3 cells. Once we click apply, we can see now our data set encompasses right around 17,000 genes and 4,600 observations or cells. We can now see a new plot that came up that shows the genes with highest expression or fraction of counts per cell within our data set. We can now move on to the next step. The next step involves a quality control step where we filter out cells with high mitochondrial gene content. Cells that contain high mitochondrial gene content are often apoptotic or already dead, and these are cells that we want to exclude in our analyses. So click on the QC tab on the left-hand side. You see the QC filtered by mitochondrial content. We simply click on the tab, and this analysis step shows up at the bottom, or at the top, sorry. It will turn blue to signify that we are on this step. Notice that graphics from the previous step in this analysis can be found at the bottom of the page below the current analysis step we are working on. Now, the default setting here is to exclude cells with greater than 5% mitochondrial content. We're going to go ahead and increase this default setting from 5% to 20% because within this data set we know that hair cells are a little bit unhappy and have a little bit of higher mitochondrial content. So to keep those within our analyses, we're going to go ahead and bump the mitochondrial content from 5% to 20%. And we simply do that by changing the filter by percent from 0.05 to 0.20. Once we have done that, we can go ahead and click on plot. We can see that 3 nice violin plots show up. The first violin plot shows us the average number of genes per cell within our actual data set. The second violin plot shows the number of raw reads per cell within the data set, and we actually also see the percent mitochondrial per cell within this data set. We can see that most of our cells are indeed within that 5% threshold of percent mitochondrial content, but we do have these cells that are higher than 0.05 or 5%, which are most likely our hair cells. And we want to keep those, so that's why we bumped up our percent mitochondrial content from 5 to 20%. Once we are happy with this, we can simply click on Save these genes. In the next step, we're going to identify highly variable genes. So click on the tab next to the highly variable genes down here at the analysis steps. Again, it will turn blue, and the Identify Highly Variable Genes analysis step will show up at the top. This is the first step in using dimensionality reduction methods to look for structure in the data. Before we perform additional analysis steps, we want to reduce our list of approximately 17,000 genes to a smaller list of genes that is most likely to reflect important variability between cell types. We're going to select the top 2,000 highly variable genes for this demonstration. And also for this demonstration, we're not going to change any of the other default settings. But keep in mind that these settings can be tweaked to get a better result at the end of your own analysis. So to get the top 2,000 highly variable genes, we simply type in 2,000 into the N top genes box here. Once we have done that, we can go ahead and click on Plot. We can see that gEAR went ahead and found the top 2,000 highly variable genes, and we can see dispersion plot of the normalized and non-normalized data here on the right, with our highly variable genes colored in black. Once we are happy with this, we can go ahead and click on Save these genes. The next step in our analysis pipeline is the principal component analysis. Again, to open up this section, we simply click on the tab next to principal component analysis. Once we have done that, we see PCA comes up at the top. Now, the principal component analysis indicates components, in this case genes, that contribute to the largest variability of gene expression. The first principal component, or PC1, has the largest effect in dividing the data set into subtypes of cells or samples. As an example, the cells in the cochlea can be divided in the broadest terms into epithelial and non-epithelial cells. A group of genes that is highly expressed in epithelial cells compared to non-epithelial cells, or vice versa, would therefore contribute greatly to the variability within the data set in that first and second PCs. In this example, we will choose two genes to colorize the principal component analysis. The first gene would be epicam, which is an epithelial marker, and the second gene to colorize is PAL3F4. PAL3F4 is an otic mesenchyme marker, which is the largest cell type, the largest non-epithelial cell type within the cochlea. We can go ahead and click Compute and Plot, and give it a second to run. We can see that our PC plot shows up here on the right. If we go ahead and click on this PC plot, we get a enlarged version. And we can see that our epithelial, or our epichem positive cells, are clustering nicely on the left, while our PAL3F4 positive, or non-epithelial cells, are clustering nicely on the right. We can also see the variance ratio within each of our PCs by looking at the elbow plot here. So if you would like to look at the actual gene list within a particular principal component, you can type in the principal component you would like to look at here and press Plot Top Genes. For this demonstration, we're going to go ahead and skip this step. So the next step in the analysis is to generate a T-Sne/U map. This step is used to visualize cells that are similar to each other. Cells that are closely related to each other will form clusters. So to get to the T-Sne/U map, we simply click on the tick box next to T-Sne/U map. There are a couple of options here that we need to fill out. In this demonstration, we want to begin looking at hair cells and supporting cells. So we will choose to colorize two genes in the plot. First gene will be PAL4F3, which is a hair cell marker, and the second gene is SOX2, which is a supporting cell marker. Now, second, option, or the second setting, is number of neighbors. The number of neighbors will have an effect on the minimum cluster size in the next step. We will choose n equals 5 neighbors for this demonstration. Keep in mind these settings need to be tweaked for your own analysis to get the best result downstream. The n number of dimensions refers to the number of principal components used to create the dimension reduction. In this example, we will utilize the first 16 principal components, and we came to this 16 by looking at the elbow plot and looking at where it starts to flatten out, or where the variance, adding more PCs won't really add more variability to the actual analysis. Again, the number of principal components can be tweaked for your own data set. For this demonstration, we're also going to be only looking at a T-Sne. So we can go ahead and unclick on the UMAP setting and click on T-Sne. Once all these settings have been put into the correct boxes, we can go ahead and press plot. This is the longest step of the analysis, but it really should only take about 20 to 30 seconds. There we go. So now we can see two T-Snes on the right, one for PAL4F3 and one for SOX2. We can go ahead and click on this to enlarge it. And we can see that indeed on the first T-Sne, which is labeling PAL4F3, or our hair cells, see that our hair cells are nicely clustered here at the bottom. You can also see that there are two SOX2 positive clusters. We have one here and one here. Now this completely makes sense because within the cochlea we know that not only supporting cells are SOX2 positive, but also glial cells. So in the next couple of steps, we can start to understand which one of these clusters is actually our supporting cells and which one of them are our glial cells. Go ahead and exit out of this, move on to our next step. So the next step is Leuven clustering. Leuven clustering is used to find the most likely groups of associated cells in a network. In the plot generated during the analysis step, each group of cells will be color-coded. Again, the number of neighbors chosen in the previous step may influence the clustering and the number of clusters produced. So, for example, the smaller the number of neighbors, the larger the number of clusters, and vice versa. So to get to the step, we simply click on the tab next to Leuven clustering. We can see that one option shows up within this analysis plot, and that's resolution. The resolution determines how granular the clustering will be. It is set to 1.3 by default. To decrease the resolution, you simply drop it to 1, for example, or increase the number for higher resolution. For this demonstration, we have found that a resolution of 0.1, which I'm going to type into the box, is a very nice starting point and gives us nice granularity. We can go ahead and press plot. And what we're going to end up having is a T-Sne. It looks similar to this, but instead of gene expression colored, we can actually see that each cluster is colored now based on gene expression. We can see that indeed our PAL4F3 positive cells, or our hair cells, are nicely colored in pink, or cluster 6. We can also see our two SOX2 positive populations here in green and red, or cluster 2 and 3. So in the next step, we are actually going to start to figure out which cluster, either 2 or 3, is actually our supporting cells. So to look at the next step, we can simply click on the tab box next to Find Marker Genes. Now, marker genes are the top genes that are highly expressed in each cluster. This will help us determine the identity of each cluster. So for this demonstration, we're going to go ahead and look at the top 5 genes in each cluster. So to do that, we can simply put in 5 and press go. We can see nice tables with the top 5 or 6 genes within each actual cluster, and you notice that you are comparing that particular cluster, in this case 0, if we look at the first one, versus the rest of the cluster. So these are the most highly expressed genes within cluster 0 versus every other cluster in the data set. If we scroll down, we can see a nice table was generated for us for the top 5 genes per cluster. We can see that our cluster 2, which we already know is our hair cells, based on PAL4F3 expression. We can see the top 6 genes here are PCP4, which is a hair cell marker, SMPX, which is also a hair cell marker, SIB2, and PAL4F3. So we indeed know that cluster 6 is our hair cells. If we look at cluster 2 and 3, which are either our supporting cells or our glial cells, we can see that cluster 3 expresses the glial cell marker PLP1. We also see that in cluster 2, it is supporting a supporting cell marker, TACT-A. So now we can start to identify that maybe our glial cells are within cluster 3 and our supporting cells are within cluster 2. To further visualize this data, we can go ahead and click on these top marker genes for these clusters that we're interested in. So for hair cells, we can go ahead and look at PCP4. For glial cells, we can look at the glial cell marker PLP1. And for supporting cells, we can look at TACT-A. If you would like to download this fine marker gene table, you can go ahead and click on the download button here, and it will put it into a nice Excel file for you. Let's say we also want to look at a marker gene that's not within this table. We can simply add it to this box below. Let's go ahead and look at SOX2, for example. Once we have done that, we can go ahead and click on Visualize. And this brings up two nice plots at the bottom. The first one is a dot plot, looking at the expression of these 4 genes that we clicked on or put into the marker gene visualization box. We see SOX2 is expressed in cluster 3 and 2, like we've seen before. PLP1 is just expressed in cluster 3, indeed showing that this is indeed our glial cells. Cluster 6 is our hair cells, or PCP4 positive cells, and cluster 2 is our supporting cells, or TACT-A positive cells. We can also see the same information, but in a Violin plot. Again, you can enlarge these by clicking on them. But we're going to go ahead and skip that for now. Now that we know which cluster is which, we can scroll down and we can see that a new table showed up within the Leuven clustering step. This new table allows us to actually change the labels within our data set. So instead of cluster 2 and cluster 3, we can change our cluster 2 to our supporting cells and cluster 3 to our glial cells, and also our hair cells, which is cluster 6, to hair cells. Once we have done that, we can go ahead and click on Rerun with Labels. Now this is simply just going to label within the T-Sne, the supporting cells, glial cells, and hair cells. You can see the glial cells are now labeled, the supporting cells are labeled, and the hair cells are labeled. The clusters that we have not identified yet simply are labeled with the top marker gene for that cluster. Now that we have done that, we can move on to the final step of the de novo analysis, which is to compare genes within clusters. So the fine marker genes is comparing a cluster to all the rest of the clusters within that data set. But what if we want to just compare 2 clusters? We can go ahead and do that by using this step. So let's say we want to compare hair cells with the drop-down menu, we can go ahead and click on hair cells. And in the second drop-down menu, we want to compare hair cells to supporting cells. We can go ahead and click on supporting cells. And let's take a look at the top 5 genes with the hair cells and supporting cells, or differences between hair cells and supporting cells. And we'll go ahead and keep the method and p-value correction method the same here, but know that we do have different methods for this. Once we have identified all our necessary comparison settings, we can go ahead and click on Go. You can see 2 really nice graphs here, but the most interesting here is the split Violin graph. So this is hair cells versus supporting cells. And we can see that these 5 genes are much more highly expressed in hair cells versus supporting cells. We can also, if we go down, look at supporting cells versus hair cells. And we can see genes that are more highly expressed in supporting cells versus hair cells. To take a look into this further, we can actually download the table of these genes. So if we simply click on Download Table, an Excel file shows up. We can open up this Excel file, and we see within each column we have names of those particular genes that we were looking at. We have the log2 fold change for those genes, comparing them to supporting cells. We have the p-value, and then we have the corrected p-value, or the FDR. And that is kind of the end of the single-cell RNA-Seq workbench de novo analysis portion. But we can take this a bit further and actually show you the power of gEAR. We can go ahead and select our top genes within our hair cells versus our supporting cells. We can right-click and click on Copy. Once we have done that, we can go back to the gEAR homepage by simply clicking on the gEAR icon here. Now I wanted to look at these genes within different data sets in gEAR. To simply do that, I can click on the gene search box, right-click, and press Paste. Pasted our top 6 genes that we just showed from the split plot of hair cells versus supporting cells into the gene search box. We can click on the exact match, and we can also choose a profile. So we are going to choose the single-cell RNA-Seq Kelly Lab 2020 profile. Once we have done that, we can go ahead and click on Search. Now this will bring up the gene expression of these particular genes within the different developmental time points of the single-cell RNA-Seq data sets that the Matt Kelly lab generated. Just to reiterate, the de novo analysis of the single-cell RNA-Seq data set that we just did was at postnatal day 2. So say I want to look at these different genes and their expression at different developmental time points. For example, let's look at SIB2. We can see, if we look at the top right, we can see SIB2 is indeed a deafness gene in both mouse and human. We can also see SIB2 expression within these different developmental time points. We have E14 here. We can see that SIB2 is expressed in inner and outer hair cells. And E16, again, we see similar things, inner and outer hair cells. We can also look at P1 and P7 expression. So this allows us to not only analyze our data and look for marker genes or differential expression between clusters, but it also allows us to take our analysis or our genes that we found to be true and compare that to other data sets within gEAR. And that's kind of what makes gEAR so powerful, is to allow you to have access to all these data sets at the same time. And with that, I would like to thank you for coming to the de novo single-cell RNA-Seq analysis breakout session.